So gene mapping that describes the methods they are used to identify the locus of a gene. Locus means place where that gene is placed, and the distance between two genes or various genes it is there. The gene mapping can also describe the distances between different sites within the gene. Example, one gene is coding or having information about multiple activities. So what these activities are, at what place they are. We have seen that the portions are there, centromer, telomer. Okay, so exactly what during or P and Q we have seen portions are there on the chromosome. So in that, where that particular information is being put in that form, that is there. So why we do it? It is to place the collection of molecular markers. So we call them as a molecular marker, or these are the various signals or information that gives about their respective position on the genome. They come in all form. They can be various genetic code, it can be various proteins, it can be various identification markers, and these will be able to give you what kind of information is present over there. So genes can be viewed as a special type of genetical markers in construction of genomic map, and these are mapped in the same way as any other markers which are present. Now this mapping is there, this genetic mapping is there along with both, that is physical mapping also. And there are two distinct type of map are used by these scientists. One is called as a genetic map and other is called as a physical map. Both maps are collection of genetic markers and the gene locus, but they are done in separate way during with a different way, and they are based on the genetic linkage information. The physical maps, they use actual physical distances which are actually which are usually measured in number of base pairs. That means how many base pairs. If you remember earlier, we have seen that nucleosome, they are having 126 base pairs. That means these are the one after another. One A is 1, suppose T is 2, G is C, C is 4. So this one sequence has got, this is 4 base pairs are there then. So like that, what's your number of base pairs are there in that particularly sequence. So these are there, that is how the distance is calculated. The physical map could be more accurate representation of genome. Genetic maps often offer insight to the nature of different region. Example, distance may show that how much away, how far away particularly gene or codons are for particular enzyme or particular structure is there. But the actually the this one genetic map will able to show you, the physical will show the distance, while the genetic will show that what are the activities which are going on there, and particularly gene rich area versus the gene poor regions of the gene, because genome can be having gene rich as well as gene poor areas, and from that we can able to identify that whether it is there, whether it is from gene rich or gene poor area is there. Now the gene mapping is done with various identification of marker using various cloning techniques, and many various research were there. Particularly maize was used then in this animal science. Drosophila is a sort of fly that has been used, and these are all giving various ideas what is there, what kind of chromosomes are there and what kind of genes are there. So it is a way to identify exactly which chromosomes has which type of genes, and what is the functionality of these genes, what exactly pinpointing where the gene lies in that particularly that particular chromosome. And mapping also act as a method in determining which gene is mostly likely to recombine based on distance between two genes. Suppose two genes are coming together to make what's your activity, or what we can get phenotype. So genotype is information and phenotype is a product. Gene is just like, means genotype is just like giving some coding or some information to computer, and phenotype is just getting a printout of it, or seeing that things is happening. So this is what genotype is there, or genotype is something like a software and phenotype is the outcome of the software. So it is a distance between two genes is measured that is known centimorgan. That is, this unit is a centimorgan. And what centimorgan is? Centimorgan is a distance between genes for which one product of meiosis is 100 is a recombinant. That is, out of 100, this one, how many are the meiotic products? You know, meiosis, in meiosis, two haploids will come together and there can be crossover, and how there is a growth of this, that is in cell division you might have seen. Particularly meiosis is a process, and after meiosis zygote is formed and then it goes for mitosis. Then second one is a physical mapping. So genetic mapping is done, then physical is done. Physical is done in what way is, there are various techniques by which the DNAs are broken down, either by using restriction enzymes. They will be, this restriction, now its name indicates that they will be restricting the DNAs or breaking the DNA into restricted portions. Maybe example, some will be able to make it 30 base pairs, 40 base pairs, 60 base pairs, like that. So these enzymes will able to break it into it. They will cut into that pieces with perfectly, just like if you remember first year anatomy physiology, and various enzymes, peptidase enzymes are breaking it, various proteins into smaller one, or various carbohydrates, or like starch, etc. They are broken into small pieces by various enzymes. So similar like that, these enzymes will be there, they will be cutting it, or they can be done by physically using the techniques of, that is sonication. So this sonication will cause ultrasonic waves, and because of that the ultrasonic waves will cause the cutting of the DNAs. Now once these are cut, these fragments are separated by using electrophoresis. I hope you know electrophoresis, you might have studied it somewhere in analysis, but it is something chromatography like situation. Whatsoever you have studied in chromatography earlier, or maybe in analysis you may be studying. It is what, it is a modified version of this chromatography. There is a gel is there, normally it is agarose. Agarose is a sort of carbohydrate. Whatsoever microbiology you might have studied agar, so similar to that is agarose. It is a carbohydrate that forms a gel, and in that gel various fluids are there, it is made, and on that gel our components are put, either genetic material, DNA, RNA, or protein, and accordingly they are called as a western blot, northern blot, southern blot, like that. So these are electrophoresis, and the current is applied. On the basis of current, depending on the current, it will pass towards either anode or cathode. Means positively charged will go towards the negatively charged electrode, and vice versa. So these are the electrophoresis, and then this separation is there. Various markers are there, just like in chromatography you will see that various marker or staining reagents are there. They will most common in that is ethidium bromide. So that will be there, that will give fluorescence, and then you can take the photograph you can see the separations. Now from that separation you can take out this DNA migrated which are there, that is called as a DNA fingerprint. So this pattern of DNA migration is used to identify what stretch of DNA is there in that particular portion in the clone. Cloning is making a copy of particular genetic sequence. We are going to study it after just this section. This is called as a clone. And once these fingerprints are analyzed, then whatsoever contigs are there. Contigs means what? It is a set of overlapping DNA segment that together represents the consensus region of the DNA. So it is overlapping DNA, and they are having similar type of activities, and from that this contig, this overlapping sequence data is there, that is taken out, that is checked, and accordingly this determined. It can be done by various automatic machines, it can be done by manual means, they are known as a pathfinders, and these overlapping DNAs are identified. There are many mechanisms nowadays, because they can be made efficiently sequence the clones to determine the DNA sequence. We have got so many machines. If nowadays during all this COVID you might have studied or seen this, what is PCR, what is RT-PCR. So there are many such machines are there which will be giving, and this physical mapping will be done, that where the genetic location is there and what are at what these locations are there. Now these mappings will be helping us to understand what kind of, in which chromosome what kind of genetic material is present, and on that basis we can have various determination, or we can have various ideas about that. Now we shall see the impact or its difference with the genome sequencing. Genome sequencing we have seen earlier. It is mistakenly referred as a genome mapping, but it is not actually the case. The process of shotgun sequencing was done, means the breaking was there, sequences were studied. Shotgun was a technology, it was the first technology. Thereafter so many technologies came, and these technologies will be there, they will be resembling. Actually it is not resembling, means only process is resembling, but the outcomes are different, but the reason for which it is done, they are different. So they resemble the process of physical mapping, that also causes the shattering of the genome into small fragments. It characterizes each fragment and then puts them back together. More recent sequencing technologies are drastically different. The scope, purpose and process are totally different, as I told you. It can be viewed as the ultimate form of the physical map, and just like each location will be given some information about what kind of activities are happening at what place. Now why we do it, why this genome map, this mapping we want to do it. So here we identify the genes which are responsible for some activities. So if you remember in our earlier lecture, just 3 lectures back, when we were discussing about various chromosome and chromosome numbers, and we have seen that various chromosomes are having their sites of various diseases. So that means that can be identified. Now we have mapped this. If you remember the figure, almost a tree like figure, that is chromosome number 1, chromosome number 2, chromosome number 3, chromosome number 4, like that. So all these chromosomes are having, now we will reach to that chromosome and we will see that at what place, at what location that particularly what we can say, the place is there, or locus is there about that particular gene which has got a problem. Now this will give new insight about the problem or new insight about the disease. So this mapping of the gene is the first step for the identification of a gene which is responsible for either function or malfunction. Okay, if we understand function, then we understand malfunction, or vice versa. And it is usually starting point of the important downstream studies. Then we understand, once we map it, then we understand that this is the gene, this is the portion responsible for this kind of activity, and because of that, then what can be the next situation. The process to identify genetic element that is responsible for disease is also referred as a mapping, and the locus in which the search is provided is considered is a constrained one, because it is not a clear idea about it, and it is derived from investigation of disease manifestation in the large family or genetic linkage from population based genetic association studies. So example, we say that hereditary disease is there, that some people has got hereditary problems, then the mapping will help that in which chromosome, which kind of place the problem is there, and why that there is inheritance is there. So this is how gene mapping is helping. Next into this is gene cloning. What gene cloning is there, and what we can see into it. So what is cloning? So it is a process of producing individual with identical DNA. That means it is a copy, same copy of the DNA which was there in the parent one, or artificially, either naturally or artificially. In nature, many organisms produce clones through the asexual reproduction. In example, bacteria, we know. Bacteria, they divide and they produce clones, means the daughter cells are the clones. In biotechnology, we call cloning, that is process of creating clones of organism or copies of cells or DNA fragments, that is we call as a molecular cloning. If we just DNA fragments we copy, then these are called as a cloning. So this term clone means copying, okay. That is that means to the process where a new plant it can be created from the twig, that is called as a cloning. And this cloning is there to have the similar molecule. Even it was used in plant biology also, but now it has been very popularly used in only genetics or biotechnology activity. So natural, I told you, reproduction is there, plants, fungi, bacteria, they are doing it, and they have their own naturally doing it, is a sexual reproduction wherever it is there. Then there are clonal colonies are there, they reproduce, and similar activities are there. There are many plants are there, you put some part of the plant into soil, and then from that bark, from that stem, the new plant will come out. There are such many plants are there, and they will doing it. Molecular cloning is actually the technique by which we are doing it artificially. It is commonly, if you remember, if somebody, you can search, it was a dolly sheep which was known as a cloned animal, where the genetics were put into, and it was a live one, that is in the live, all the DNA from the mother as such, it was put into the dolly, and therefore it was a cloned animal, it was called as. So this cloning refers to the process of making multiple molecules. Cloning is commonly used to amplify DNA fragments which is present in the whole gene. It can also be used to amplify any DNA sequence like promoter sequence or stop codons or whatsoever it may be, or non-coding sequences. Any kind of portion of the DNA is amplified, or it is multiplied, or many copies of it can be made easily, if we say. So it is used in wide area of biological experiment and practical application, ranging from genetic fingerprinting. So this fingerprinting, you know, just like our fingerprints are our identities. You go for Aadhaar, you go for something, you go for many offices, you put your thumb, then that fingerprint is recognized, because it is totally different than, or it cannot be resembled. It is same, it is your own specific one. Similarly, here genetic fingerprinting is like that. Fingerprint, here each genetic one is a different one. Means maybe body we are looking same, maybe functionality we are looking same, or even we say that hereditary some diseases are transferred from one generation to other generation. Still genetic fingerprinting will show that at certain places, at certain sites, there are typicalness of that particular genes will be there, and that is copied as such into the next one. That is actually the cloning is there. The term cloning is mistakenly used to refer the identification of chromosomal location. So it is not there. Therefore, we have seen it before that. Identification of chromosomal location of a gene, we call it as a genetic mapping. We have seen it as a. Okay. So in positional cloning, this is done, mapping is done first, and then positional cloning is there, that only some portion of the, once we identify that this is the gene, we mapped it, then we identify that this is the gene, and then we go for positional cloning. The localization of gene to chromosome or genetic, it does not necessarily enable one to isolate or amplify the relevant genomic sequence. The amplification of any DNA sequence in the living organism, that sequence must be linked to origin of replication, and then whole replication activity is to be done. Okay. So these are there. We have got various vectors are there, which are called as a cloning vectors. So these are the small piece of DNA into which the foreign DNA can be inserted, and this This is very important, cloning vectors, because this cloning vector idea is now taken into the gene therapy. So one after one we are going. We have seen what are gene, we have seen how they are doing their job, what are the peculiarness of that, we have seen it, we have studied it, we have studied how genes are responsible for various functional or functionalities, and then how we identify various gene at what position, what kind of thing is there, we have seen as a gene mapping. Then next our step is to identify what genes are or which genes are responsible for what kind of phenotype. After that we understood that, or we can understand that what kind of diseases are linked with the diseases, and then next we are, once we map it, then we can have the cloning vectors, that is the small piece of DNA into which the foreign DNA is inserted, and these vectors they will exist, that will allow protein production and affinity tagging, and accordingly RNA DNA production can be done, and host of other molecular biology tools can be done. So it involves 4 stages, that is fragmentation, that is break apart some portion from the DNA, ligation, that is bind it or join it, suppose in a whole big molecule of thousands of base pairs. Now I need base pair numbers, or they are important, then base pair number may be 37 to 87 I have taken, then 120 to 160 I have taken. So these all are fragmented, they are separated, and then just like intron exon we have seen earlier, then they will be broken up, they will be separately done, and then they will be brought together, we will call it as a ligation. So we are gluing or sticking together pieces of DNA which we need, and that will be in a desired sequence. Then transfection, whatsoever this we have done now, we have made what, we have taken the fragments, these were at other places separately away from each other. From mapping we understood that which portion we need, so from mapping we have taken it, then we have fragmented, as I told you, maybe 37 to 87, 120 to 160 and so more. So all these fractions which were there, which were important, we have taken together, and there we have ligated together. Ligation is, enzymes are there, we call them as a DNA ligases. They will be binding these together, they will be sticking them together and make, now this has become a different sequence that it was there. Then this is what we wanted and we got it. Then we will transfect it to this DNA into cells. So it can be any cell, it can be microbial cell, it can be animal cell, and when we transfect it, then now this has become the DNA of this cell. Okay. Then screening selection out that cell which are successfully transfected with the DNA. So in whatsoever cell we have put into it, we have inserted this DNA which we have artificially made now. This artificially made means we have made sequence we have made artificially. So this artificial sequence we have put into that DNA, and we check that which of these cells have got it, they have got this new DNA, and they have considered it as its own DNA. Now once they consider it as its own DNA, then they will process it for all these processes including transcription. Now this cell will be able to do the function what you desire. Okay, this type of thing is called as a recombinant technology. That is, earlier DNA was there, and now you are adding the piece of DNA into it, and this DNA will be doing the job which you assigned, because you know that this much portion of DNA is responsible. So initially the DNA of the interest needs to be isolated to provide the DNA segment of the suitable size. Then ligation has to be done. Then you have to put it to the vector, whatsoever DNA is there which is able to carry it and which should be able to incorporate it into the other DNA. Then next is the vector which is frequently circular is linearized using again enzyme restriction enzymes. Restriction enzymes are the enzymes which are actually hydrolyzing enzymes, okay, like DNases. But just like you have seen dipeptidases, tripeptidases, if you remember earlier your digestive system studies, okay, or this carbohydrate metabolism enzymes. So from that dipeptidases will make it to 2 peptide each, or tri will make 3 peptides, means 3 amino acid sequences. So like that, here this will be having restriction enzyme will be making it at particular length, maybe 10 base pair, 20 base pair, and therefore we call them as a restriction enzymes. So they are incubated with this, and then we put it with the enzyme DNA ligases. They will be specifically able to join with the vector, and they will one after another they will be doing it. So this is all, this is therefore very complicated study. Genetics is, and therefore it is always put into, or it is always combined along with the computers, and therefore that becomes a new branch, that becomes a bioinformatics. So this number of alternative techniques are available. They are called as a chemical sensitization of cells, electroporation, optical injection, biolistics. So these are various techniques are there which are used for. We will not go for techniques because again we are not geneticists, but these transfected cells are cultured, and then this cultured they are artificially developed. If those who are interested can go for can search about Dolly sheep, which was almost 15-20 years before it was made as a cloned animal, first clone animal. And here cloning means basically when the zygote level it becomes, then the DNA you incorporate into that will sticker it successfully, and the animal grows into a full animal in the womb of mother. It is not in the test tube. Then that is again put back into the uterus, and then it will grow like that. It was there. Will not just those who are interested can go for extra knowledge how that Dolly sheep was made, how the cloned animal was done. So these are various techniques they are done. There are many ways by which you can do it, either unicellular or multicellular animals are there. Then cloning stem cell is nowadays being considered, that is somatic nuclear transfers are there, so that if the stem cell can be cloned, you can make the copies of that. You know, stem cells are very important. Many of them are pluripotent, that they can give rise to many cells. So where the cells are not proper one, in that these cells, if we can have the cloning of that, and we can insert this as, so we call them as a research cloning or therapeutic cloning. And in the people where maybe there are deficit of one or other kind of tissues, in that this cloned stem cell we can incorporate, and we can see that whether new tissue is formed wherever this kind of genetic problems are there. Okay, these are the benefits of this. It is done by creating embryonic stem cells in the hopes of treating disease. Actually, this has not become much successful, but there was very big hype. Let me tell you, in one of my very close circle, there was a boy who was suffering from cerebral palsy. That is a very debilitating disease, mentally and physically. Nothing can be done. It is just life, but without anything, without much inputs and outputs. Physical means no walking, no movements like that. People say that there are now stem cells are available, and it takes means they were charging lot of money for that. Somehow people go for it, but it has not become that successful. But yes, there is a one branch where it is going, that this can be done. Embryonic stem cells will be taken up, particularly in the treatment of diabetes, Alzheimer's, or as I told you, this cerebral palsies, etc. There are in many diseases are there. So in case of someone with the Alzheimer's disease, the nucleus from skin cell of that patient is placed into empty egg. Then reprogrammed cell becomes to develop into embryo, because egg cell react with the transferred nucleus. This embryo will become genetically identical to the patient, because it is from the same cell or skin cell of the same patient. This embryo will then form a blastocyte, which has the potential to form to become any cell in the body. So since other neural cells cannot be developed, but these blastocytes can develop into the neuronal cells, and if we can inject it into this brain of the Alzheimer's patient, it can take the place, and this can be there. This is what thinking is there. So far no big success is there, and many things are there. many reasons. This is one way of cloning, one function of the cloning. Then particularly this was done with the human cells. It can done into the farm animals, and then farm animal will able to produce the we can say that the factories of organs or the tissues which are there. And then instead of using sperm and egg genomes to replicate, this oocyte will be inserted into the donor's somatic nucleus. So instead of human body, then animal bodies can be used to produce this cloned tissues, and thus it will become a cloning technique can become a very important, that is called as a somatic cell nuclear transfer, that is SCNT. So this will become, or this is expected to be popular one for many things. Good method for producing agriculture animal for food consumption. So without much going for sexual reproduction and all, if it is it successfully clones sheep, cattle, goats and pigs. So far it has been done. Another benefit of this cloning is seen as a solution to clone endangered species. If somebody of you have seen the film like Jurassic Park, then it shows that somebody found the egg of these dinosaurs, and they put it to laboratory, then they clone it, they put it to other animal, and they develop so many dinosaurs. Some of them are good, some of them are dangerous, some of them are very devastating. So this has all information about the cloning, how this cloning is there and how this cloning is there. I told you about clone chip Dolly. Here you can get the information. That is how the clone, it was born after 277 eggs were used for this cloning technology, and it created 29 viable embryos. Out of these, 3 of these embryos survive until birth, and only one survived to adult one, that was called as a Dolly. So those who are interested can see it. That was continuous observation by the scientist for each stem, and then it was put into the uterus, and it was allowed to develop like that. So this was all stories about it, various stories. There are many scientists are working on, many laboratories are there, private laboratories are working in belief that once we can find solutions to the diseases which are so far not treatable, because neuron damage are there, maybe Alzheimer's and many other neurological deficits and many other such diseases. So if we can make it, it will be useful. Similarly, as a donor cells, mitochondria that contain own mitochondrial DNA, they are left behind, and the resulting hybrid cells, they are retaining into mitochondria structures. And the clones such as Dolly that are born from, they are not perfect copies of the donor of the nucleus. So this is the biggest problem, because mitochondrial DNAs are separately present in the mitochondria, and they are not there. So these are the various ways. This is organismal cloning, that is or asexual reproduction, or cuttings from the plants or vegetative reproduction is done, that is particularly done in the horticulture field, and from that various activities are done. Then various organism can be artificial clone. That is first step was the German embryologist awarded Nobel Prize in Physiology for discovery of how the embryonic induction various parts are there, and this reproductive cloning used as a somatic cell nuclear transfer to create animals that are genetically identical. This process makes the transfer of nucleus from donor adult cell, that is somatic cell, to the egg, and from which the nucleus is removed. The cell will be or the cell from the blastocyte cyst can be taken from the nucleus, and then insert various DNAs into it, and that will be developed into clone methods. So already I told you about Dolly sheep, and what are various things were there. It was the first mammal to be cloned from adult somatic cell, and this was a very big example, and from that because it led to the animal farming. It was done, I'm sorry, I told you 20 years, it is almost 1996, almost 24, 25 years back, and it died in 2003. So this was there, but there was a problem into that. I told you mitochondria is the problem because mitochondrial cells were not be there. How the cloning was done, you can see that how this it was done from the mother to this one, and how the dolly was formed. So these are there. Now we can have so many species cloned. You can see the list of animals that have been cloned. Because of this, we have got so many technologies nowadays for nuclear transfer, and every day no many techniques are there. So these are many such animals are there. Now out of this zebra fish I can show you. This zebra fish is animal which is used nowadays particularly for many of the pharmacological studies, for the study of various drugs on the instead of using mice and rat and other animals. Zebrafish has become a popular animal. Sheep, I told you, mice is there, and in mice you can have so many varieties of mice, rhesus monkeys, pigs. So these are rats. There are various are there. Then human cloning is also being done. That is for the creation of genetically identical copy of human. The term is generally used to refer artificial human cloning, and this is particularly getting or making available delivery of identical twins. But there are many controversies, ethical issues are there, because this is not correct to do it, and if the person can have, particularly human cloning was done for one more reason actually, that if one brother, one of the children was having a problem or genetic disorder, to correct it, the another this what we can say, fertilization was done, and from that fertilization one copy of the cell, the pluripotent cell, was taken and can be inserted to that. So this is discussed type of theoretical human cloning. They are known as therapeutic cloning and reproductive cloning. Therapeutic cloning would involve cloning cells from human for use in medicine and transplant, which is still an active area of research, but not in medical practice anywhere in the world. So far it has become successful. Two common methods of therapeutic cloning are being researched are somatic cell nuclear transfer and pluripotent stem cell induction. These are there. But as I told you, there are many issues are there for medical one, many legislations are there to make this, and at many places it is not legal. Cloning extinct and endangered, just now I told you about dinosaur and about movie Jurassic Park. If you go for this article, you will find that how those who have seen Jurassic Park, you can see. Those who are interested can see that movie also, Jurassic Park. There are many versions of Jurassic Park are there, and many are there. There are many species they become extinct. Extinct means because of they cannot survive the adaptation changes, or maybe excessive hunting of this particular species or killing of that particular, some of the animals. They are become extinct. They were there earlier, but now they are not there. They cannot be regenerated. So accordingly, the projects are made to save this those which are now. Suppose only few animals are remaining, then the people go for that. That is called as a cloning. So there are many such things are there. Many cloning are there. Many novels are written over there. Many we call it as a fiction, means what we call it as in Marathi, fiction means that is, there are many such fiction movies are there, many fiction novels are there, and on that basis this cloning is used as a what we can say, as a method. There are many clones are there, and these clones are used as a warriors, as a factories for organs, anything like that. So there are many are there, and if you again I'll remind you that if you remember COVID-19, and if you'll read about it, you will find that people say that it has been made artificially, many people say that there was, I am not able to recall now, there was a book and there was a movie where it was put into that somebody will make this kind of virus and it will be put into. So these are all things are happening because of the cloning, and this cloning can making as a it can be beneficial, it can be harmful. As I told you, cloning can be there for warfare uh, at many places. It can be put into, many people can use it, as I told you, and it can make various bacteria, it can make various viruses, and accordingly movies are made for that, to show that how cloning can be anything. It can be very beneficial, it can be very harmful, it can be anything in between. So there are many things which are there for cloning, and but this cloning is put into practice, or this cloning is useful in what our next topic we call it as a, that is called as a gene therapy. So once we understand, important thing is from this is, once we understand that now we have went step by step. We have seen that there are chromosome, in chromosome there are areas to what we call as a gene. These genes are responsible for having certain information which is responsible for normal functionality of cell, normal structure of cell, that is structure and function, to what we call as a basic unit of organism. So that is there, and this structure is basically, once we understand, then we mapped it at what place what it is there, and then next we go for can we make it, can we separate some portion. We try that was scientists separated out, then they tried to, can it be copied. Yes, it was copied. Can it be inserted? Yes, it was inserted, to what we call as a cloning. Once there was a cloning, this cloning was successful, and even animal was made out of it, and accordingly one after another sets of things were available. Now next into this is, now can we use it? Yes, the gene therapy was the next step. We can see this gene therapy in our next class. So far whatsoever you have understood, you can go through it. If you have any difficulties, please let me know. You can put it on group.